Gene editing systems have opened up a host of new ways to explore how life works, and also have the potential to help improve human health, produce better crops, and fight environmentally damaging pests. Here, 3 young scientists from the University of Otago's Department of Biochemistry tell us about how they plan to use CRISPR gene editing in their diverse research projects. We're not going to show you exactly how CRISPR works. There are many great explanations already available on the net. Instead, you will learn about some of the ways CRISPR is being used in research right now.
Well, my name is Soledad. You can call me Sol. Come from Buenos Aires, that's the capital of Argentina. I just got a grant to start studying the circadian rhythms in legumes, in a small little legume that calls Medicago truncatula. Okay. So lentils are a legume, peas are legumes. They're actually a really big source of protein for humans, and it's very used for livestock, for feeding livestock, especially alfalfa, for example.
Sol wants to know how legumes adapt their circadian clock to the environment they grow in. One day we'll be able to use this to make plants better adapted to the new environments that climate change will bring.
And I measure something called leaf movement. So plants move even though we don't see them. So when you film the plants during continuous light, you will still see them opening and closing their leaves. But they will have their own rhythm that is set by their internal clock. What I'm specifically looking for is one change in a nucleotide in the whole DNA that can help me relate how the plant grow there and their genetics, particularly looking at the circadian clock. Once you measure all this leaf movement compared to the genomic and try to find responsible genes for this difference that you see, you try to mimic these changes using CRISPR-Cas gene editing. The plant that has A and change it to C with gene editing and see if changing this gene will make the plant behave like the other one that has a C. We put it in the plant using tissue culture techniques.
Sol and her assistant Shireen put the CRISPR editing machinery into new plants using a special kind of bacteria, agrobacteria, that transfers DNA into plant cells. They cut some Medicago leaves into pieces and mixed them with agrobacteria that have the CRISPR machinery encoded inside them.
And you incubate it with the bacteria is able to go inside because of the cuts that you make to the leaf. And that's leaving them with bacteria.
They then put the pieces onto nutrient-rich cell culture plates. Infected cells start forming calluses on the side of the leaf pieces.
So each of these squares that you see here are the explants that we took. That when they are in this media, they start developing calluses. Can you see the little white? That's embryos. And then it's a little less humid. There, do you see that from the explant there is a little, you need to focus into there. There is a little plant.
Eventually, the calluses start growing tiny plants with the altered gene inside them.
So you get these plants that are ready to be moved to the soil because they have long roots.
Very cool.
So this was a leaf.
That was a leaf.
Once you try this and you actually see this, you can then translate it to other legumes. So you could find the gene that is homologous and try to see if when you change this gene.
I'm Gemma McLaughlin.
I always get my lab mates to bring in any wasps they see. Yeah, and you can just store them in the fridge. These guys I use for mating purposes. So we collect all the queens from a nest at the end of the season.
It's a jam
It's a jam jar. Yeah, I use these when I see queens at my house. I just put them in a jar and bring them into work. Yeah, they just go to sleep and they just sort of stop.
For months?
Yeah, they can. Well, the queens hibernate for months. Usually everything else just dies out. Invasion biology has always been my thing, and it's a big problem in New Zealand, as we all know. And then it's like, well, wasps are everywhere. They came here accidentally. It was just hibernating queens in World War II plane parts. They've just done so well here that it's quite detrimental to other native species.
So you're getting rid of wasps
genetically.
by
Genetic methods.
genetic methods?
Yeah.
Gemma and her supervisor Peter Dearden are looking into how to make a gene drive to eradicate invasive wasps from New Zealand. A gene drive consists of some bits of RNA that can alter or silence a specific gene, plus the genes that encode the CRISPR-Cas machinery. The genetic modification uses CRISPR to insert and spread through a population at higher than normal rates of inheritance.
So yes, they've done it in mosquitoes, so they just had to inject a female, and then the effects they wanted were expressed in all her offspring.
Inside the cells of an organism that inherits one copy of the gene drive, the whole drive copies itself onto its partner chromosome, so that the genome now has 2 copies of the gene drive. In this way, the change is passed on to up to 100% of offspring.
So if I did that, that would be really effective, because then I could just inject a queen and have all her offspring do what I want, as opposed to having a queen injecting every egg she lays within the small window before they develop too much as well.
So you're targeting, the cells that make the eggs rather than the eggs themselves.
Mm-hmm. So I'd like to target this gene called double sex, which is like sex determination in wasps.
To begin with, Gemma will visit a lab in the USA to learn the techniques they are using to introduce this system into bees.
What I'd like to do is the method called non-homologous end joining, which is pretty much you make a double-stranded break in the DNA, you cut out a section and then you join the ends back together. And for that, I want to target this exon on chromosome 3, and for the double sex gene that produces males. And so just cut that out so it produces a stop codon instead. So a stop codon means that's the end of translation. We stop making proteins from here. So then it doesn't make males at all. So eventually then you're only having females, they run out of males to reproduce with, and so they eventually die out.
There are many obstacles to overcome before a gene drive could be possible for wasps.
It's a bit harder with wasps because they're not a model organism and we don't have their genome online accessible. We're working on that now.
Oh really? It hasn't been sequenced yet?
It has, just by some postdocs in the other room over there.
Oh, excellent! We need to understand their fertility genetics better and learn how to grow wasps in the lab.
Okay, so this is the first layer of a wasp nest. So this is the pistil here that they attach to sometimes trees. They're usually underground, but that's the first layer that the queen makes all on her own. If you flip it over, it's just little cells. They have really small cells at the start because she's just trying to produce as many workers as she can. And once she's got enough, then they take over and build the rest of the nest, and then she just becomes a baby factory.
But most important of all, we need to look at all the benefits and hazards that a gene drive could bring, and decide as a country whether this is a path we should take.
It does have multi-generational effects, and the biggest fears is if it would escape to another country. It's a valid concern. It's something that should be looked into. I mean, we shouldn't just release a whole bunch of genetically edited wasps into the wild, so we are going to look into things.
My name is Oluwatobi Iboda. I go by Toby for short. So originally I was actually born in Nigeria, but then my family moved to the United States when I was real young. I actually played rugby in the States for about 10 years, which is why I knew of New Zealand in the first place.
So you've decided to do a PhD with Steph?
Yes.
Dr. Stephanie Hughes?
So I'm part of the Neurodevelopmental and Disease Lab.
The neurons?
Yes, I would assume so. Now, my project within her lab is the first that looks at a gene that is associated with Parkinson's disease, actually. I'm studying this gene called ATP13A2.
What is it called again?
ATP13A2.
ATP
13A2. Or PARC9. PARC9 would be much simpler.
Some genes have terrible names. The PARC9 gene encodes a protein that's found in the part of a cell called a lysosome. Lysosomes are like the recycling centre of the cell. Waste molecules are sent there to be broken down and reused.
When we have defects in this particular organelle, like especially in the neurons, like we see a lot of neuronal death, and it's been something that's been a feature of Batten disease, like something that we're noticing in Alzheimer's disease as well, and more recently Parkinson's disease. Yeah, so there have been published research on this gene showing that mutations lead to the development of a juvenile form of Parkinson's disease. We see development around age 12 and deteriorating rapidly over the next few years, and it also presents with dementia. I'm using CRISPR-Cas in order to make my model.
First of all, Toby needs to grow some brain cells, neurons, in the lab. But you can't just take someone's neurons out of their head and start experimenting on those. So Toby has another way of getting neurons.
So I'm using these induced pluripotent stem cells to create my model.
What does that mean?
So these...
Where did they come from originally?
Were originally harvested from an individual. They were actually fibroblasts-like cells, so skin. Now pluripotency means that it has the ability to differentiate or become several different types of cells. We harvest the fibroblasts, might treat them to turn them back into their pluripotent state, and we call those the induced pluripotent stem cells. And then through certain treatments, we're able to direct their differentiation process or their changing process into a particular cell type. In my case, neurons. These are my cells here. I'm growing these iPS cells on these 10 centimeter plates. These cells I will then be inducing into neurons later down the line. These colonies might contain many different cells that are isolated and have distinct edges. So yeah, these are our colonies of cells all the way over here. You usually want to harvest these cells and either begin the differentiation process, which I probably will be doing today.
Once Toby has made his neurons, he can use CRISPR to change the PARC9 gene in them and look at how it changes the way the cells look and behave.
There are several different versions of CRISPR-Cas. There is the CRISPR-Cas, the normal system, which knocks out my gene. So I can introduce another copy that carries the mutations that we see in this gene that lead to Parkinson's disease. And then there's the CRISPR-I, which is a adapted system which turns off expression of the gene. So I'm able to knock down the gene without actually cutting or affecting the DNA underneath. So the next step then is to begin to characterize my model. Are they similar to what we're seeing in Parkinson's disease patients? And if it is, therefore the model becomes more relevant and can be used for further experimentation in order to develop treatments in the future for Parkinson's disease.
Would you be thinking of trialing things like gene therapy?
Yes, so that will be down the line as well, like once we have confirmed the model.
Are you still playing rugby?
Oh, not as much, Mike. You guys have been playing rugby out of the womb. I can't compete with that.